Students, the topic we are looking at today is mutation, and we are starting right now. Good day, everyone. This time I'm talking to you about mutation. M-U-T-A-T-I-O-N. What is mutation? And what are the types of mutation? And what are the effects of mutation? Those are the things we're going to be looking at in the lesson today. Mutation. The arrangement of nucleotide sequence in the DNA is highly conserved, and this does not change over generations. Under certain conditions, however, there may be an alteration in the nucleotide sequence in the DNA molecule that may be heritable in the offspring. Under this condition, a mutation is said to have occurred. A mutation is said to have occurred. So a mutation can be defined as an alteration in the molecule, in the nucleotide sequence, in the DNA molecule. And that change is heritable. For the sake of those who don't get the gist all along about what we're saying about nucleotide sequence and changes, you see, I already spoke to you about recombination, whereby you break a pencil, some pieces are lost. That pencil initially had a nucleotide sequence. I'm using the pencil as an example, had a nucleotide sequence arrangement. Now, the process of breaking and recombination, some pieces have been lost, some pieces are added. Those pieces, we are talking about nucleotide sequence. Those small pieces that are added have affected the codon, have affected the base pairs. So, what happens is that there will be a change. The change may be mild. In the nucleotide sequence where you have U or where you have T, all of a sudden you now have C in that place. It will change the nucleotide sequence, if you understand what I mean. Mutation is therefore an alteration in the nucleotide sequence in the DNA molecule that becomes perpetuated in a heritable manner in the genome of an organism. Cells with altered genotype are called mutants, while the original type of organism with the normal genotype is called the wild type. Mutations occur in all generations, in all organisms, beg your pardon. However, the probability of mutation occurring spontaneously is very low. For example, in a mature culture, in a culture of bacteria containing 10 raised to power 12 cells, that is about some trillions or so of cells, the probability of a mutant in such cell is only 1. So you can see that mutation is rare. Mutation is, however, higher in plant and animal cells. There are some agents that affect DNA, hence increase the rate of mutation. These agents are known as mutagens. It could be physical or chemical in nature. Examples of mutagens include ultraviolet light, X-rays, gamma rays, HNO3, bromouracil, and some other chemicals. Because of mutation, that is why when you go to take your X-ray, they tell you not to stay too long in the room. The person taking the X-ray wear a special type of cloth so that the ray will not penetrate the individual. Now we'll go to look at types of mutation. Mutations can be grouped or categorized into 3 major categories. These are, number 1, mutation at the level of DNA. Then number 2, mutation at the level of the organism. And number 3 is mutation at the level of the gene. Let's quickly talk about mutation at the level of DNA sequence. Different types of alterations can occur in the DNA sequence as a result of mutation. The most common is the point mutation. This is the type of mutation whereby only a single nucleotide is affected in the DNA. This may be as a result of replacement of a nucleotide by a different one. A point mutation could occur whereby a purine nucleotide is replaced by another purine nucleotide, or pyrimidine, or another pyrimidine nucleotide. Such is classified as transition. It is called transversion when a pyrimidine base is replaced by a purine base. A point mutation could also be as a result of insertion or deletion. The consequence of point mutation in a DNA molecule depends on whether that section is a gene or not, and also on the functions performed by that gene. Now, I will talk to you about mutation at the level of the gene. Whatever type of mutation occurs in the DNA molecule will probably have effect if it occurs in the coding region of the gene. These could have several consequences which could result into silent mutation, missense mutation, nonsense mutation, and frame shift mutation. What is silence mutation? Silence mutation is a gene. Silence mutation in a gene will occur if a point takes place but does not affect the coded for by the new codon. This will not give rise to a mutation that has phenotypic expression, e.g., UUA and UUG code for leucine. So if the A in UUA is replaced by G, which becomes UUG, or UUG becomes UUA, it is silent because the two are coding for the same thing, which is leucine. Now we go to missense mutation. It's a point mutation that results as a result of codon that also decoded for whether not this will cause a mutant phenotype depends on the precise function of the mutated amino acid in the structure and function of the protein. For example, such as an active site of an enzyme is likely to have more effect on the function of that enzyme rather than if it occurs in other parts of the enzyme. So this is missense mutation. Number 3 is nonsense mutation. Is said to occur if a point mutation, that is a codon specifying, let's say UUA leucine, into a termination UGA. UGA is termination. So nonsense mutation is that to make nonsense of the nucleotide sequence. You have UUA leucine. Now a mutation have occurred, UUA becomes UGA, which is a stop codon, which is a termination codon. So the reading frame will stop at that point. So whatever is after UGA, which was formerly UA, will not be able to be read again. So definitely that will cause a drastic change in the reading frame of the nucleotide sequence. It will change it drastically. The result will be a truncated gene codes for a polypeptide that has lost a segment, resulting into a mutant phenotype. Number D is frame shift mutation. Is a consequence of an insertion or deletion of a nucleotide. You know what insertion means? You know what deletion is? Insertion means you have seven students somewhere. When they insert a student, you have maybe in the project group you are 7. They now inserted a student. They sent another student to the group, 1 student. It becomes 8. It has inserted. But you are talking about nucleotide sequence here. Now you have codons and the arrangement already made. When something is inserted, it will affect the arrangement that has been made. The same thing applies to deletion. Let's say you have 6 students or 7 students. Now 1 student is removed. It's removed from the student. It will affect the reading frame. When a mutant is caused by insertion or deletion in a gene, an extensive type of generic damage may result. The consequence of such mutation is a destruction in the reading frame, not vianding, in the reading frame of the gene, and hence the sequence of the polypeptide that it codes for. This will begin at the point of the gain or loss of the nucleotide. Hence, a totally different sequence from the point of mutation will result. In most cases, biologically inactive gene products and mutant phenotypes are produced as a result of frame shift mutation. Now we talk about mutation at the level of the organism. To produce a mutant phenotype, the nucleotide sequence therein must give rise to the mutated gene, whose product is unable to fulfill its normal function in the cell. Cells which are unable to tolerate the loss of function will die. Such mutations are called lethal, L-E-T-H-A-L. That means something that leads to death. Lethal mutations. In both prokaryotic and eukaryotic organisms, some mutations will inactivate proteins that are not essential to the cell, while others could result to proteins produced, reduced, or modified activities. Some examples of such mutations in organisms include conditional leader mutation, orthotrophic mutation, and antibiotic resistant mutants. So we'll look at conditional leader mutants. Mutation could occur in unicellular organisms that leads to the production of conditional leader mutants. This type of mutant is sterilized by surviving only at a particular set of conditions when cultured in a medium. The most common of conditional leader mutants are those that are temporarily sensitive. In this situation, the mutant is only able to survive at one temperature range, and temperature higher or lower than that range will lead to the death of the mutant. That's why I said to be conditional. So if a particular condition is changed or modified, it will affect the mutants and in a later way. The next one is orthotropic mutants. This is a type of mutants that lacks in gene production involved in the synthesis of an essential metabolite, such as an amino acid. This type of mutant can only be kept alive if the metabolite is supplied as a nutrient in the culture medium. For example, a tryptophan autotroph of E. coli is a mutant that lacks one of the enzymes involved in the biosynthesis of tryptophan. Hence, such mutant is unable to survive in a medium that lacks such tryptophan. That lacks tryptophan. The third we want to look at is antibiotic resistant mutants. Antibiotics will naturally kill wild-type bacteria, but they have effect on the resistant mutants. To such antibiotic. This results when the target of the antibiotic become altered. An example of such mutants is the streptomycin-resistant mutant. Streptomycin exerts its effects by interfering with protein synthesis in bacteria. Streptomycin binds ribosomal protein and thereby preventing messenger RNA translation. Consequently, no protein will be synthesized in the organism, and this will lead to the death of the bacterium. In streptomycin-resistant mutant, the gene for the ribosomal protein is mutated. This leads to an altered ribosomal protein and is still able to fulfill its role as ribosomal protein but can no longer bind to the streptomycin. Consequently, streptomycin will no longer be able to affect the mutated bacterium. Quickly, we'll look at the effects of mutation on multicellular organisms. What are the effects of mutation on multicellular organisms? Different types of mutations are formed in animals and plants The effects of some mutations depend on the nature of the mutation and the types of cells involved. In plants and animals, there are two types of cells. These are the somatic and sex cells. A lethal mutant in a single somatic cell may be unnoticed since cells in tissues and organs are constantly replaced with new cells. A mutation that affects the somatic cell may have an effect if the mutated cells give rise to progeny cells that also carry the mutation. Such effects in plants are easily observed in the variegated colors on the leaves of such plants. In man, a mutation in the somatic cell will only be important if it confers on the cell some dangerous properties, such as the ability to divide in an uncontrollable manner, which may lead to cancer. On the other hand, a mutation in the sex cell will be passed into the offspring through the egg or sperm cells, such that all the somatic cells of the offspring will be mutated. Many heritable enzyme defects leading to inborn errors of metabolism have been shown to be due to gene mutations. Examples include albinism, sickle cell anemia, galactosemia, phenylketonuria, alkaptonuria, and many others. We'll quickly talk about galactosemia. Galactosemia is the presence of galactose in the blood. Galactosemia is a hereditary or heritable disease due to lack of enzyme involved in the metabolism of galactose. It is a disorder of infant with inability to metabolize galactose of the milk lactose. This normally results into growth failure and cataract formation. Albinism, phenylketonuria, and alkaptonuria. These are diseases of inborn errors of metabolism, principally due to single gene mutations. They are all associated with the metabolism of an amino acid phenylalanine. In some human beings, the metabolism of phenylalanine can be summarized as shown below. Talking about albinism specifically now, albinism is a genetic disorder characterized by lack of pigmentation. It is due to deficiency in tyrosinase enzyme that converts into melanin. Then phenylketonuria. It is also a genetic disorder characterized by mental retardation in infants. It is due to deficiency of phenylalanine hydroxylase that converts phenylalanine in the blood, which may be subsequently excreted. Alkaptonuria, also a genetic disease or disorder arising from metabolism of phenylalanine. Although it is not a serious disease, it is not characterized by excretion of dark to black urine due to oxidation of homogenic acid in urine. It is as a result of deficiency in the enzyme involved in the conversion of homogenic acid to melilacetoacetate. The accumulation of homogenic acid in the blood leads to its excretion in the urine when exposed to air, and is oxidized to a black compound. So we're going to continue from here very soon. We'll just take a break and I'll be right back. Welcome back. As I said earlier, alkaptonuria or black urine disease is a very rare inherited disorder that prevents the body from fully breaking down two protein building blocks, that is amino acids, called tyrosine and phenylalanine. It results in a build-up of a chemical called homogentic acid in the body. So this alkylproteinuria is characterized by excretion of dark or black urine due to oxidation of homogentic acid in urine. I'm going to talk to you soon.
That mutations can be bad or it can also be good. Mutation can occur during DNA replication if errors are made and not corrected on time. Mutation can also occur as a result of exposure to environmental factors such as smoking, sunlight, and radiation. Often, cells can recognize any potentially mutation-causing damage and repair it before it becomes a fixed mutation. Mutations contribute to genetic variation within species. Mutation can also be inherited, particularly if they have a positive effect. For example, the disorder sickle cell anemia is caused by a mutation in the gene that instructs the building of a protein called hemoglobin. This causes the red blood cells. The subject of mutation for now by giving you an assignment. I'm going to group you. The assignment will be done in groups. So you do the assignment and then you submit it in the first week of January. The assignment is on Ames test. AMES, Ames test. How does Ames test link up with mutation? And then the procedure for carrying out the Ames test. I want to give you details. And as you look for details, tell me how it has been done right here in Nigeria, and some workers have done the Ames test. So it's an assignment to be in a group, and then you submit in January. So as you go for the Christmas and holiday New Year break, you do the Ames test and then you submit it in January. Bye-bye and happy Christmas and happy New Year in advance. See you in January. With sickle shaped and leads to sickle cell anemia, where the uptake of oxygen is not enough to sustain the individual that has such sickle cell diseases. Another effect or bad aspect of mutation is the formation of cancer cells. Cancer cells are products of mutation. So these are some of the bad effects of mutation. Put the subject of mutation for now by giving you an assignment. I'm going to group you. The assignment will be done in groups. So you do the assignment and then you submit it in the first week of January. The assignment is on Ames test. AMES, Ames test. How does Ames test link up with mutation? And then the procedure for carrying out the Ames test. I want to give you details. And as you look for details, tell me how it has been done right here in Nigeria, and some workers have done the Ames test. So it's an assignment to be in a group, and then you submit in January. So as you go for the Christmas and holiday New Year break, you do the Ames test and then you submit it in January. Bye-bye and happy Christmas and happy New Year in advance. See you in January.